welcome to the next module, antifreeze protein part 2, ice crystal binding and its mechanism. So before we look into ice crystal binding, let us look at what are the different types of ice crystal planes. So here is the structure of hexagonal structure of the ice is given here. The bottom one is called the basal phase, and the one here in the rectangle in the side what is shown is the prism phase, and this is the c-axis, and this particular one is called the secondary prism plane. So these are all some fundamental things we need to know before we get into what are the different types of ice crystal planes and how the ice crystal formation is prevented by AFPs. When we study about the mechanism, we need to know this basic information. So there are four major things we will be actually looking at. One is the primary prism plane, that is the one which is facing towards the person. If we look at this is the ice crystal, and if this is the hexagonal ice crystal shape, the one on the side here is called the prism phase. So this is the primary prism plane or the alpha plane. And then we have a basal plane which is at the bottom. Base is what is the one. It is both at the bottom and the top it is present. So this is the basal phase or the basal plane. It is called as the C plane, and the height of this is given as a C-axis. And we have the secondary prism plane. So this part between the two corners is called the secondary prism phase. And we also have the pyramidal plane that will start from here to here, and then it will grow till the top. So that one, when we look at the actual structures, we will see. So what are the descriptions with respect to the AFPs? The prism plane is the fast growing side face of the ice crystal. This is aligned with the A-axis of the hexagonal lattice. And what is the growth characteristics for this particular plane is there is a rapid expansion along the A-axis contributing to overall crystal size and the shape. This is how generally the ice crystals grow. And when we have the AFPs slow or inhibit the growth along these fast growing planes. So ice generally grow very fast in this prism phase, and when AFP binds to these places, they stop the ice growth. So that is how AFPs function. And when we look at the basal plane, this is a flat slow growing base of the ice crystal perpendicular to the C-axis. So this is the basal one which is perpendicular to the C-axis. And here the growth is basically slower expansion compared to the prism phase. So prism phase grow very fast, basal phase grow very slow. And when we compare the AFP's action, AFPs strongly inhibit the growth on the basal plane also. And when we look at the secondary prism phase, this is the secondary prism phase. So here the additional phase that forms at the secondary angles to the primary prism phases. So this is one primary phase, this is another primary, there is another primary. So all the hexagon sides are called the prism phases. So between two prism phases, this is what is called the secondary prism phase. And this is less prominent than the primary prism plane, and they contribute to side branching only in the case of ice growth. And when we look at the AFPs actually interfere with the growth of secondary phase, causing irregularity in the crystal growth. And when we look at the pyramidal plane, which we will see soon, it starts from the base of these two things and go till the top. So this pyramidal plane, this phase that forms at the specific angles between the basal and the prism plane, giving a pyramidal shape. So basal plane and the prism phase, they form here like a triangle they form. This is a slow growing phase that contribute to the overall symmetry of the crystal. AFPs actually slow or modify the growth, creating more symmetrical crystals. So this is what is the actual four planes on which the crystal growth actually happens, and how AFP actually prevent them from growing. So here is the schematic representation of the one we have been talking about. Let us start with the fast growing or the prism phase. So this is the prism phase on which the growth actually takes place. This is the fast growing axis, this is the A1 axis, and this is the A2 axis. And then we also have the basal plane. Base is the base, that is the top one. So this is the C-axis, from the bottom to here is the C-axis which is going up. And the basal plane pattern is actually shown here, how the ice crystal growth happens. And then we talked about the diagonal one, which is the secondary prism plane. So that is given in the green color here. And then we also have the pyramidal one, that is also shown here, which is an angle happens between the fast growing prism plane and the basal plane. So this is the triangle like what we have seen in the pyramidal one. So there are different types of AFPs. If you look at DAFP, so they actually prevent the growth by binding to the basal plane pattern. The pattern is actually given here. We will look a little bit deep in the next slide. We also have the secondary prism pattern and AFGPs, that means antifreeze glycoproteins actually stop the ice growth by binding to the primary prism plane, and AFP3 generally focuses on the pyramidal plane. It binds to the pyramidal plane. Let us look at the types of AFP binding to the ice planes. We have 4 different type of growth, and when we look at the fast growing pattern, here the ice expands rapidly along the fast growing crystals, which we have actually seen, and how the AFP affect is they basically slow or inhibit the growth along this one. So this is what we have actually seen. And when we look at the basal plane, AFP inhibit the growth by leading to a distorted or inhibited crystal growth. And when we look at the pyramidal plane, AFP interfere with the fast growing phases, promoting slower, more stable growth along the pyramidal plane. And when we look at the secondary prism pattern, where here AFP suppress or slow the growth of these secondary phases, leading to irregular crystals. So here are the examples. So we have the 4 basal plane, prism plane, secondary prism plane and the pyramidal plane. All the 4 are given here, and we can see this DAFP, which is the beetle dendroids canadensis. It can have all the 3 things. That means it can form, it can prevent the crystal growth through the basal plane or the primary prism plane or even the secondary prism plane. So all the 3 can be inhibited by DAFP1. And when we look at MP, that is Antarctic bacterium M primordiensis, this will have the same similar to the DAFP1. This also prevents the ice growth by the basal plane binding, primary prism plane binding and the secondary prism plane binding. But when we look at AFGP, that is glycoproteins, they generally work on the primary prism plane only. That is why it is shown as the yellow color. And AFP3 actually has both the primary prism plane inhibition as well as the pyramidal plane binding and inhibition of the ice growth crystals. And SSAFP, which is the short thorn sculpin fish, it has mainly the secondary prism plane inhibition is the only thing it is doing. And when we talk about the winter flounder fish, this mainly focuses on the pyramidal plane only. So these are all the different types of fishes and the action of different antifreeze proteins and how they prevent the crystal growth. There are few models to study the ice growth inhibition. Model A is proposed by Riemann and De Veer's concept. The second one was called as a Felting concept. These are used to explain the inhibition of ice growth. So we can see here, this is figure A and this is figure B. So in the figure A, this focuses on the inhibition of stepwise growth across the surface. So that is how here the prevention takes place. And in the figure B, this emphasizes the prevention in all directions. That is, it's a 3-dimensional arrangement of AFP molecules which prevent the crystal growth. Both these models support the concept of AFP lowering the local freezing point and causing thermal hysteresis. We will be studying about what is thermal hysteresis soon. But in figure B, which is having a more 3-dimensional structural arrangement, here it is a more comprehensive and the 3D mechanism for ice growth inhibition, and this makes it the most likely to be effective across various crystal orientations. So this is a much better way of mechanism by which the ice growth can be prevented. And here are the explanation about both the models. The first one, which is the Riemann and De Veer's concept. So here the AFP molecule inhibit step growth across the ice surface. So this is the simple one. So here the inhibition occurs by preventing the advance of growth steps across the adsorption plane of the icy crystal. So what is the geometric assumption that is made? The growth occurs by step advancing across the surface with the AFP spinning these steps in place. And what is the key concept? The AFP stop the growth of individual steps on the surface of the crystal, effectively blocking the lateral expansion of the ice. So this is what proposed by Riemann and de Vries. Whereas when we talk about the felting concept, here the AFP molecules inhibit growth normal to the surface of the ice crystal, that is perpendicular to the surface of the ice crystal, the growth is actually prevented. Here the inhibition is achieved by AFP spinning the growth normal, that means perpendicular to the ice surface like buttons on a mattress. So that is how they actually prevent. And here the ice surface is treated as a three-dimensional structures with AFPs preventing growth in all directions, not just along the surface. So this is the major difference between these two models. And what is the key concept? The AFP creates a mattress-like structures that prevents ice from growing in any direction, ensuring complete inhibition of the ice expansion. Let us look at the relationship between ice plane affinity and ice crystal morphology. We have three different types of AFPs, type 1, type 2 and type 3. How they actually prevent the ice crystal growth. The crystal planes preferred by the ice binding proteins are the type 1 mainly binds to the prism planes and they prevent the ice growth. In the case of type 2, they bind to the basal planes, and type 3 they bind both to basal and the prism planes. So this is the difference between type 1, type 2 and type 3 AFPs, how they bind to the ice planes. And the growth direction below HFB, when we look at, see the burst growth along the C-axis leading to the elongation along the vertical axis of the crystal. So this is how they prevent the growth of the ice crystals. And similarly, when we look at the ice crystal morphology, all of them are having bipyramidal structures, and the type 3 has both bipyramidal and triangular structures. And when we look at the effect of thermal hysteresis gap, in the thermal hysteresis gap, the ice crystal remain immobile and the growth is halted until a critical temperature is reached. This is in the case of type 1. And when we look at the type 2, the ice crystal in thermal hysteresis gap experiences inhibition of growth leading to step pinning along the basal plane. And when we look at type 3, the growth is inhibited in the thermal hysteresis gap, leading to more multidirectional crystal expansion. And here is the examples of how they prevent the crystal growth. So when we look at AFP1, and this is AFP2, AFP3 and A20L and TAS8, and AFP3 is also another type is given here. So how they bind to the either basal plane or the prism plane, and how they reduce. When the AFP's concentrations are increasing, they actually stop the crystal growth. So this is a solvent with 40% sucrose is present, and when 1.5 micromolar AFP1 is given, it reduces or it tries to inhibit the ice crystal growth formation. So here are the binding motifs of the AFP to the surface of ice crystals. These are present in various fish and other species. Ocean pout has this particular structure in a three-dimensional one. So these are all the small proteins with a flat surface that is rich in threonine, that is colored in the light blue here, which binds to the surface of the ice crystal. So this is the main area where the ice is actually bound to these AFPs. So here we have fish, that is ocean pout and the winter flounder, and we have another three proteins from insect, that is yellow mealworm beetle. So this is from an insect, and the budworm moth, Spruce budworm moth is also given. Another one is the snow flea. So these three insect and the two fish have different binding motifs for AFP. So how they are binding is actually given here. So the ice binding site is relatively hydrophobic and flat, and these yellow highlighted regions are the place where the ice is actually bound. So the AFP1 has all alanine rich amphipetic alpha helical polypeptide, and if it is AFP2, they have disulfide bond rich globular protein. So this is like a circular the globular shape. This is a linear one. And when we look at the AFP3, it has the twisted loops folded into triple stranded beta sheets. So this is the area where the ice actually binds to this particular protein, or the AFP binds to ice crystal structure. And this is the defective isoform of AFP3, that is A20L, that is alanine is replaced with lysine. So this binds only weakly to ice crystal surface, and we have the fungal AFP. This has this particular area, it has a 7 ladder, 1, 2, 3, 4, 5, 6, 7 ladder beta helical structure as shown here. So these all ice binding sites are relatively hydrophobic and they are actually flat. If you look at this is flat, this is flat, this is also flat. All of these things are flat and they are also hydrophobic. So in these areas only ice come and bind.